cell line encyclopedia. There's different data sets for if you've got protein expression like mass spec, interactive virtual environment. Project Achilles is for synthetic lethalities, etc. There's a whole lot of places to look for data, and honestly, it can be kind of dizzying trying to understand where you want to pull this from. So in order to try to help make that better, I just want to go through some of the most common places that people download data from and how to easily access them. And do a step-by-step with you guys today on your laptop so that you're comfortable doing so. So the Cancer Genome Atlas is one of the biggest data sets available in terms of cancer sequencing data. It's got over 2.5 petabytes of data, and just to put that in perspective, even though we don't really use DVDs anymore, that would fill up almost 200,000 DVDs. These data cover 33 different cancer types, including 10 rare cancers. There's over 11,000 patients of data there, and then 7 different data types, so including expression data like messenger RNA and microRNA, germline and somatic mutation calling, and copy number alterations. Has anybody downloaded data from TCGA before? All right, great. So we're going to do that. So let's all go to portal.gdc.cancer.gov. And so TCGA is now housed in this thing called Genomic Data Commons. All right, everybody there? All right. So the first thing you're going to need to do is click on data. Makes sense. That's where we would expect the button to be. After that, we are going to go ahead and select some different specifications about what kinds of data we're looking for. Is everybody on this screen? Nope? Okay. So everybody on this screen. Let's see, try Googling Genomic Data Commons. Oh yeah, that should be it. Yep, the top one, GDC. Let's see what the exact website is I have up here. So again, the exact website that you should have is portal.gdc.cancer.gov. So portal.gdc.cancer.gov. Okay, perfect. Got it. Portal.gdc. Yep, that should be it. That is. Trey, you guys find anything? Okay, and then everybody else got it. All right, perfect. All right, cool. All right. And I do not know why your computer is not letting us. Maybe it has a problem with Safari.
Anyway.
Oh yeah, we can maybe have you buddy up with something.
No worries, I got it, yeah.
Okay, perfect. All right, so the next thing you're going to want to do is just click the data button. All right, after we click data, you can see there's all these different options we can choose from. So on the left here, we can select the primary site and also the disease type. So let's say you're looking for kidney cancer. Right over here, you can see on the top, you can press kidney, and then if you wanted to select instead by disease type, you can also go here. You can select by different projects, so that is where the initial data source was, who generated it. I guess they also have a data set from Target as well. All right, so for the purposes of today, why don't we go ahead and just select kidney cancer, and then see what that pops up with. And then once you do so, go ahead and press enter. Any problems with that so far?
Just kidney cancer?
Oh yeah, let's start with kidney cancer. The important point here is that whatever cancer you're interested in, just select it here. And then after that, we're going to be able to select, to further stratify the data we're interested in through these different different pie charts. So if there's any other filters you're interested in applying over, you can also apply those as well on the left. So once you do that, we can select the kind of data that you're interested in selecting. So if you actually hold your mouse over one of these circles, you can see what kind of data is underneath it. So in this case, you can't see it on my screen because it's no longer the website, but I believe this is the transcriptome. But whatever data set it is, just go ahead and hover over it, and then you can click on it. All right, has everybody clicked on one of those? And also, are the pie charts slowly getting so that more and more of them are the same color? Good. That's just representing, like, as we're drilling down into more specific data. All right, and then after we select that, there should be some number of data sets left. And then after that, you can just download it. So let's see what everybody's screen looks like. Okay, good. Okay, good. Yeah, so this is saying there's one data set that we're going to download.
All right.
All right, good. Everybody.
I'm actually looking for a project for my research question.
So I wouldn't worry about that one. So this would be if you want to specify like where the data came from, like whether or not like we generated it Sloan Kettering or wherever. Instead, let's select, let's say we want to do RNA-seq. We'll just select that. And so then you can see there might be still lots and lots of files left. So at this point, once you drill down into the amount of the data set you're actually interested in order to download it, all you have to do is click Add to Cart, and then download. Now, I would not actually download the data. At this point, you might have several terabytes of data selected, and that will, might be bigger than the storage space available on your local machine. But the moral of the story here is that TCGA and Genomic Data Commons has a lot of data available to download. If you're interested in trying to compare your study to somebody else's existing study, the way to do it is probably access their data. This is one way to do so. Download it and then analyze it yourself. But there are some caveats, so you need to make sure you're actually downloading what you think you're downloading. For instance, if you're only interested in an accounts table that might be, I don't know, a couple kilobytes, maybe 500 kilobytes or a couple megabytes, you probably don't need to also download the gigabytes of BAM files that come along with it. And the interface isn't the most user-friendly system that has ever been designed, so it is a little bit, it does have a bit of a learning curve in terms of figuring out exactly how to access the data once you get there. So the next website that I want to go over is something called the Gene Expression Omnibus. A lot of times in papers you'll see somebody else say like, oh, upload it to Geo. And what they mean is all of the authors, when they had their data, had to upload their actual data set to this website. And so that means that tons and tons and tons of data are available through Gene Expression Omnibus. And we're going to go over exactly how to access that. There are two major types of Geo datasets available. There's the Geo datasets section and also the Geo profiles section. So the Geo datasets is a study level database where users can look for actual entire studies that they're interested about. So let's say you want to look up a study that has RNA-seq of prostate cancer. They'll have manually curated a list of things that are involved in RNA-seq and prostate cancer. Versus the Geo profiles page is focused on specific genes. So let's say you want to see how p53 is involved in prostate cancer. You can go ahead and use Geo profiles in order to do that. So let's go over to the Geo datasets. So why don't you Google NCBI Geo, and then we'll all get onto the web page. Good, and then you're going to click Geo datasets. All right, perfect. Everybody on there? No problems? All right, great. Yep, and then it should say your datasets over Gene Expression Omnibus, and then after that we're going to want to do Geo datasets. There should be a button up here for selecting everyone your data sets over geo. There we go. Search for geo. There we go. All right. Now that we're at geodatasets, all you're going to need to do is just enter the terms that you're interested in using. I would caution here that, at least in my initial trying to use this function like a Google-like search, I had difficulties. So I would probably recommend opening up the advanced options, where you can have a more granular control of exactly what you're looking for. And also specify the terms in the way that the search algorithm is hoping to see. So why don't we go ahead and click on Advanced Search, and then y'all can pick your favorite search terms that you're looking for. Yeah, so, or the name, say we want to do, so you can see if we go to like all fields, if you highlight this right here.
All fields.
Yep, so if you go down there, and then you can see all of the different options.
Here.
Yep, so exactly, so you can see all the different fields that you can specify. And again, so this is just to show that, so if you have a specific paper that you were reading and you want to get this data set, you can just go here and type in the GeoAccession number, and then that data set will pop up immediately. Or if you're interested in looking for RNA sequencing or DNA sequencing of prostate cancer, you could specify, I want studies that are about prostate cancer and also that have, also are ChIP-seq, or for whatever your favorite protein, or for whatever your favorite ChIP-seq study is. All right, so overall, again, I know once you know which buttons to push, the website is not too bad. The key is just understanding which buttons you need to push in order to get to where you want to be in. The key thing is getting here and then going to your advanced search.
Sometimes you can get similar information just by clicking on the gene NIH, or blasting sequence or whatever, and it gives you the position of the genome, the references. It gives you a lot of information.
Yeah. So this is more about trying to get other people's data sets or at least geo data sets is about gathering other people's data sets and then downloading them for your own use. But yeah, there's definitely other places where you can get information about specific genes. Great, and then you can see that when you actually do the search, there will be your data set name, a description of the data set, what kind of data it is, what organism it's in, who generated it, etc. You can also look for a specific gene of interest within a given data set. So if you click on one of the first data sets, you can actually search and see, all right, if I'm interested in KRAS, we can see, or in the next slide, I think actually I have P53, we can see how that gene is actually up or down in a given data set. So you can actually crack open the data set without having to download any of the data. So why don't we do that? All right. Any issues with taking a look at the individual gene? Okay, we can click on it, then you should be able to search. Okay, so you click on an individual study, so say this is the first one. Then you scroll down. Oh, is this not it has to be a data set, so it's not one of the data sets. There's no, so one of the things to note is that in order for these features to work, it actually has to be one of the curated data sets. Let's just look at this prostate, and this, and then you'll be able to search for, I guess this is microRNA, so I don't have it. Later on, you'd have to search for like the actual RNA sequence for the given gene, so that we would have gene level information. But the upshot here is that you can look for a given gene of interest within one of these geo data sets without actually having to download it and use whatever your favorite processing software is. And what this allows is for you to check the expression of a gene of interest very quickly and relatively painlessly within a given data set. All you need to do with that, click on a gene, and once we click on a gene that we're interested in, we'll be opened up to this screen, which will show us the relative, so it'll show us the different sample grouping, or the different expression values rather, that's in red. What the overall rank order of expression was, so for the entire, say, 10 or 20,000 genes that are available in the data set, where did that gene's expression fall? As well as which samples we had present, and also the sample class. So that is in this experiment, say you had your controls, you had your experimental condition, and here's how P53 varied between the experimental group and the control group. So it's very useful for if you just want to do some exploratory data analysis without having to do any, invest that much time in downloading the data. The website also offers the ability to do differentially expressed genes, but I would caution against using that functionality specifically. Unfortunately, the way in which the website currently calculates differential expression is just by having a specific non-multiple testing corrected two-tailed t-test, so I don't think it's the most statistically robust method by which to get differential expression data. But it will allow you to do so if you just want to see at a glance what genes are up or down. In order to do that, all you need to do is assign samples to an appropriate group. After you would click through this, and then see which genes were differentially expressed. I have these slides so that you can look through if you're interested in doing it. If you actually want to do differential expression, this is not how I would do it. One of the very useful functions is we can actually perform hierarchical clustering on the data sets all online without ever having to download the data. So let's make sure that everybody's at one of the data sets that are available. So why don't you go to the search function and type in chronic high-calorie diet effect on KRAS-driven lung cancers. Yep, so the title of the data we want is chronic high-calorie diet effect on KRAS-driven lung tumors. Just so that we're all using the same data set. And from here, we're going to work on how we can actually perform hierarchical clustering on our data. All right, everybody finding the data set okay? Nope, did the search term not work?
Yeah, I think this one was
Okay, so can you show me what the website looks like? Oh, no, so you go to the search engine, and then up here type in, you mind if I type it in? Just, what did I say, high-fat? Or want to go ahead and type in chronic high-calorie diet? KRAS, and that should pop it up. Are people having an issue getting access to this? Yeah. Nope, found it. I guess here. Let's see. Chronic high chocolate. Oh. No worries, calorie, and then that should. No worries. This is why why we want to go through this at this level of granularity, just because stupid stuff can make it so that it's very there will be one step that prevents you from getting your data, and we want to make sure everybody can at least go through through this pipeline by themselves while they're here. Anybody else having issues? Everybody else is on the access. Yeah. All right, great. So go ahead and click that. Perfect. And now we're going to go and cluster this heat map. Alrighty, so let's select the kind of clustering we're going to want to do. In this case, let's just go with an unpaired correlation and select complete linkage. Don't worry about what these mean right now. If you're interested, you can go. If you end up using this tool, I'd recommend reading through the guidelines prepared by Geo. But for now, let's just go ahead and make our heat map. You can also notice it can also actually do k-means and k-median clustering as well, but we won't be talking about that today. And then once we select what kind of clustering we want, all we're going to do is press display, and then you should get this map. All right, so let's. Yeah, no. This is exactly right, and then we're going to be able to select with that. Okay. So once you click on that,
why did you click on random? Well, that makes sense.
Oh, I'm just using this as an example data set that you can use for, you can do this with any available data set that you have in this
So
case.
randomly you pick the first
Yeah, literally, I think I went through and this might have been the one I used as an example. Or I happened to search KRAS or something when I was looking for an example. There's nothing special about this data set. But are people, has everybody accessed the heat map? Everybody's got heat maps? All right, great.
Sorry. So it's this first one here,
Yep.
and then,
So just click on it. And then you scroll down. You go to cluster heat map. Yep. And then you can go ahead and cluster heat map. All right, then if you want to, you can even zoom in on a region of interest. Let's say you notice that the samples seem to be, there seems to be a huge difference in one of the blocks of genes that have been found through hierarchical clustering. And then we can actually zoom in, say, on one region just by if you click once, that these lines right here will show up. And then after you zoom in, it'll let you see what the actual gene names are. And then also show you what the classification of the samples was. So in this case, were they high growth or low growth, or high calorie or low calorie for this specific study? And then at that point, you can then actually go search for these genes. So if you were interested in what the effect, in this case, if you were interested in genes involved in metabolism of high growth, or what happens in KRAS-driven lung tumors, we now know which genes seem to be driving a lot of the clustering. If you end up using any of this data, I also would recommend opening up the experimental design and value distribution just for quality assurance, quality control. So this will allow you to see what the average expression of a given sample was. And what you want to see here is that all samples look like they were sequenced to about the same depth, or that you had about the same signal off of each of your microRNA, or each of your microarrays. It'll also show you the sample name, and again, what the experimental design was. So, the moral of the story on this is that GEO is a huge repository